Hello everyone. Today I'll be talking about recombinant DNA and transformed bacteria. This is part 1 and I'll be posting a part 2 shortly. Before we move on, I'd like to talk about 3 terms. These are essential for the next few topics. So first is a plasmid. Plasmid is basically a small circular double-stranded DNA loop found in bacteria. It's separate from the main chromosomal DNA and it can carry extra genes. And this is important because this is the plasmid that we are modifying that we're going to insert into the bacteria later on. Within the plasmids, you can find certain genes. For example, we have our LAC-Z gene, which you should know from the lac operon. Let's say this is the LAC-Z gene. Say over here we have our AMP gene. This will be another type of gene that you may find within a plasmid. And basically, when AMP is expressed, it will confer resistance to antibiotic ampicillin. Next is restriction sites and restriction enzymes. These go hand in hand, and essentially when there is a restriction site, which is either on the plasmid or like on a gene, it acts as a site where the restriction enzyme can come and cut. And this is important because of this, via sticky ends, we can insert foreign DNA within these plasmids, which will transform it essentially. So here I drew out a nice example of what that will look like. So as I mentioned earlier, let's say there's an ampicillin resistance gene right here. We have our HIND3, which is going to be our restriction enzyme, which will be placed on the restriction site on the gene. And essentially it'll create a cut right here, which will open up the plasmid essentially. And let's say we have our foreign DNA right here. It's going to be flanked with 2 HIND3 restriction enzymes. And once they cut the foreign DNA, the sticky ends that will form, or more so like something like this, will form, and that means that the foreign DNA can then be inserted into the region of the gene that we cut with HIND3. So this will be inserted here. So usually with the insertion of foreign DNA, it causes the plasmid that contains that gene to stop working, so it can't express the resistance to ampicillin anymore. In this case, we have the same plasmid with the same gene for resistance to ampicillin. We also have the same restriction enzyme with the same restriction site on the plasmid. However, here we have the foreign DNA, but this time it's being flanked by 2 different restriction enzymes called EcoRI. It will still make a cut and create the sticky ends, but this time the foreign DNA cannot be inserted into the plasmid. This is because the EcoRI will create a different type of sticky end, which will prevent the matching of the base pairs. So that means the EcoRI sticky end will be different from this HIND3, and because of that, it can't be inserted. And since the foreign DNA is not inserted, that means that the plasmid will still be resistant to ampicillin. That's it for part 1, and in the next video, I'll be talking about how the sensitive E. coli reacts under different environmental situations when different types of foreign DNA, recombinant plasmids, and regular plasmids are inserted within the E. coli. Thank you.